So, as we know from previous lessons, enzymes are biological catalysts. So, they break down long, complex molecules into small, soluble molecules that can be absorbed by the small intestine and then are processed to carry out things such as releasing energy through respiration. So, we know that enzymes are made of proteins and they form a 3D structure. So we now need to know how are these proteins actually formed and how is this 3D structure formed. So proteins are made up of amino acids. So amino acid is our simple molecule. So we've got our individual simple molecules. And what happens is amino acids join together and they form long chains of proteins. So you've got our individual molecules joining up to form our long chain of protein. So it doesn't just stop there. We don't just have long strands of proteins. Something else happens. So we have these protein strands. They twist and they fold. And these twists and folds between the individual strands of our proteins are held together by weak forces. So weak forces are holding together our two strands, 2, 3, 4, hundreds and hundreds of strands of proteins together. So these weak forces in between. And these twists and folds form a unique shape. So this unique shape is really, really important because it means that we can only bind to one particular substrate, which we know from previous lessons, enzymes are specific to one substrate only. So when we've got these proteins and long chains of proteins, multiple of them, twisting, folding, forming weak forces in between them to create this unique shape, we then form our enzyme. And our enzyme has a specific active site. So a specific shape, and it is only specific to one substrate. So an example could be lipase and lipids. So we looked at last lesson, lip for lipase, its substrate, lipids. So this particular active site and the shape of our protein twists and folds, which are creating this unique shape, can be affected by two different things. So it can be affected by temperature. So increasing or decreasing the temperature could potentially have an impact on the shape. And the second one being pH. So pH is talking about whether the conditions in the surroundings are acid or alkali. So for example, the pH in the stomach is low, it's acidic, and this affects our enzyme activity. So just to recap, amino acids join together to form proteins. They form twists and folds with weak forces in between the molecules. This creates a unique shape. This unique shape has this specific active site for one substrate only. So your example, lipase and lipids, and it is affected by two different things, temperature and pH.